spring part of the semester in 2017. I'm really glad that we are on big numbers again. And restoration seminars are also exciting for me all the time. There are always new people coming in, and we have, I don't know if everybody received the program for this semester, but we have now the final SPOT program, and it's sent out, and it will appear on TAC's homepage, but I will send out emails, and whoever is not on the email, probably if that paper goes around, and if you didn't receive an email on the seminar, I actually will be happily put you on the list. And today, Krystal, I should say, my colleague Krystal, she volunteered to actually have the first semester, first seminar. I wouldn't say volunteered, but I convinced her to do that. And we actually have been working now together since 2015, and I think we are getting used to each other and form a good team. And so I would like to introduce Krystal. She's our greenhouse manager for the native plant program, and a great colleague. She actually has a BS in biology from the Duane College, a master's in environmental studies from the University of Montana, and she has worked in the environmental restoration industry in Montana for 16 years. That's great experience. So she, if somebody, then she would really know how to grow native plants, and she proves that really well. And she has worked at Montana Tech for 6.5 years now, first as a native plant specialist and then the greenhouse manager, which she is today. And she has been talking about the challenges of growing native plants for restoration. And I give the words to you, Krystal. Thank you for taking this first. We have cookies and we have a Coke, but we don't have glasses. And Krystal brought these things too, so pass it on.
Okay, so basically my talk is going to give you a very slight overview of the native plant program, and then we're going to go into my specialty, which is how you grow native plants, which is basically what I've been doing for 16 years. Okay, so the history for the Montana Native Plant Program is Chris Douglas, sometime before 2008, wrote a greenhouse grant because we have a lovely greenhouse now, but it wasn't so lovely before 2008. And then after we got the greenhouse grant, then Chris Douglas and her husband Rick wrote a grant that they submitted to the NRD to get funding to start the native plant program. And that ran from 2008 to 2012. And then that was, the first grant had a focus on growing native forbs and creating islands on the Butte Hill. And they did that using soil and putting the plants in that soil. The second grant, because the first one worked well, the second grant, Chris and Rick wrote the second grant also, and that one is still running. It is started in 2013 and it's going to go to 2021. And when Robert took over, a few things changed. There's now more of a focus on trees and shrubs, and there's also an emphasis on how to get rid of the Eurasian grasses, basically crested wheatgrass. Okay, so the next slide is the importance of natives, because if you're here, you probably know that natives are important. And one of the first things is what is a native? And because native can mean have a variety of meanings. You can have national, meaning the entire continent, North American. maybe not every North American species will work here. And so you can also have regional, which means Rocky Mountain or Western US. Also, that doesn't mean it's going to work well here. You can also have local, which is a lot more site specific, which means right here in Butte. And what we're looking at doing is getting local plants, not because locally adapted species like we have here are adapted to the conditions here, and they can actually survive catastrophic events, 100-year events, the stuff you don't see very often. Okay. Then what's out there? Another thing that we're looking at is doing a lot of monitoring, because we need to know what is out there, where it is, looking at a variety of factors. So what we are doing is going to less modified areas, because there aren't many purely natural areas around here anymore. So we go to the least modified areas. We do some monitoring to find out what species are there, what their habitat type is, so that when we go back on the Butte Hill, we get the correct plant in the correct spot. It's also a little bit more objective than the monitoring is an objective determination. What we're doing is not going to recreate what was once there. That's way too complex, but we can put more species in there that will create a somewhat more complex environment than just having the Eurasian grasses that we have now. Then we've got design. And by design, I mean how we're going to put the plants in the ground. We have to think about stuff like succession. Are we going to, if we do a scraping to get rid of the Eurasian grasses, that's a disturbed site. And so what's going to work best there are early successional plants, something like lodgepole, something like blazing star, Phacelia, something like that. And then there are environmental factors that we have to consider. And environmental factors, soil type. If we don't put soil down, because the Butte Hill has been so big, they've used many different types of soils because they had to get it from many different places. So soil type varies. Depth of soil varies. Elevation, where it's at on the hill, varies. And slope, how steep the slope is, can vary too. Some plants like it very steep, some plants prefer a flatter surface. And then aspect, is it north, south, east, west? And that all of those have a factor in how we're going to design and where we're going to put these species. And then, of course, looking at what they've done before is they haven't looked so much at those factors, and they've put non-natives in. And a lot of what we're seeing now is on south-facing slopes, you're getting almost 95% coverage of crested wheatgrass and not much else, which is when you have just one species like that, it's unsustainable in the long run. Okay, so now I've talked a little bit about the problems. So the problems that we're seeing are with non-natives. Non-natives can be exotic, like crested wheatgrass. It's not considered noxious or invasive, but it is exotic. And part of the problem that we're seeing with that is it creates a heavy thatch layer, and it doesn't allow any of the native species to germinate in between the crested wheatgrass bunches. Then we also have, we're looking at noxious weeds too, like knapweed. Let's see. So one of the questions I get asked a lot is, what is a weed? And it's a lot of what a weed is about perspective. my definition, and this is my personal definition, is it's any plant that's where it's not wanted. And I say this because back where I'm from Nebraska, milkweed, I consider milkweed's a native. It's a beautiful plant. It's great for pollinators. But in Nebraska, it's considered a noxious weed. And that is because Nebraska is a ranching state, and milkweed is toxic to cattle. And so it is actually illegal to plant milkweed in Nebraska for that reason. So there are different perspectives on what a weed is. Okay, so the last one is survival is not easy, and it's not so much about getting out on the hill, but when we go through this, I'll be talking about how even growing it in a greenhouse, getting it out, overwintering it, all these factors, survival is still not easy doing that. So just remember, none of this is particularly easy. Okay, so now we're going on to the stuff that I really know about, which is seed collection, seed treatment, seed cleaning, anything to do with growing seeds and growing plants. So this is some of my students collecting seeds. Okay, some of our equipment: paper bags, plastic bags, hand pruners, buckets, tarps. And I always put hands in there because actually we're a small enough operation that we don't have the big fancy equipment. So your hands are the biggest thing that we use, even though we have a lot of other stuff to help us out. I also wanted to talk about paper and plastic bags. People think that, I always get some people wanting to put seed in plastic bags. What happens when you're out collecting on a hot day and you put it in a plastic bag? Seeds still have a lot of water. They have to dry down. If you put them in a plastic bag, the seed sweats some of the water out. It starts losing its moisture. It's in a plastic bag. There's nowhere for that moisture to go. They start rotting rather quickly, get moldy. So I try to encourage everyone to put their seeds that they collect in paper bags because they're breathable. They dry down rather easily. The one exception is for berries. And then you want to put them in plastic bags because you want to hold that moisture in until you can clean them. And you have to clean them right away because they'll rot, and actually they ferment really easily. And unless you've got elderberries and want to make some elderberry wine, it doesn't work too well. The next one, then we've got some more equipment. We don't have. Pole pruners, berry pickers. The one up on top is the berry picker. The sickle down below. Berry picker is probably self-evident. You can use that to collect berries. The sickle is really great for grasses. Tennis racket, you might think, wow, that's kind of weird. But what we often do, the reason why I put tarps in the previous one is you lay the tarps around a plant, and then you can hit it with a tennis racket or some other piece of equipment, and if the seed is ripe and ready, it will just fall off the plant really easily, and then it's on the tarp, and you can pull it away, and you're good to go. I'm hoping to convince Robert, since we're going to be doing more trees and shrubs, to get a pole pruner, because pole pruners are really good for trees, especially pines, which we're going to be growing, because pine trees, the male cones are usually closer to the bottom, female cones are on top. It's so that they don't self-fertilize. So they're wind pollinated, so the male cones will fertilize the surrounding trees, and the female cones are on top. But that means they're harder to get. Anyway, so I'm going to talk a little bit about harvesting and hand harvesting, like what we do here. It is it's easy for us to do because it's local. It is more expensive. Anytime you have more people out there, people are basically your most expensive quantity. It's not equipment or anything like that. But it becomes a lot more expensive. This is Douglasia montana, Rocky Mountain Douglasia, and that is one of the plants that has to be collected by hand because the seeds sit right down in the leaves, and so you have to pick them out by hand. But hand harvesting, when it's local, you get a greater variety. Because if you go the mechanical route, it's usually regional. They are grown like a crop. It is a lot less expensive because you use a machine like that. But there's less choice because the only way that a mechanical harvest will work is if the seed sits up high enough from the plant that it's easy to combine the whole thing off. Okay, so and there's less choice because there are a lot less species that is available that mechanical harvesting will work for. Okay, so another thing that I am always thinking about is the amount to harvest. How much should you leave on a plant? Because if you collect everything, you've just destroyed the seed bank for that year. And that can be a problem because, if something catastrophic happens, you need to have that seed bank there. And if you don't have that seed bank. So usually I like to leave at least 50% of the seed on that plant. And what I do, rather than trying to just say, okay, leave 50% on, I only go back maybe weekly to the plant. Even though the seed may be ripe, I try to collect as much as I can at one time, and then I let it go so that the other seed has a chance to drop, so that if other animals need that seed for food, there's a lot of reasons to watch how much you collect. Hopefully you have enough, there are enough plants in the area that you can get the quantity of seed you need without disrupting the environment too much and the animals that depend on it. And then I also consider if you have one plant sitting all by itself or if you have a large patch. And the reason I consider that is because of genetics. If you have one plant, it's pretty isolated, or if you have a really small patch of only a couple, you're not going to have as good genetic variation. So it's always if you have to, you do it, but it's always preferable to go to a large patch or have numerous plants in a smaller area, just because you're going to get better genetics, you're going to get better survivability, you're going to get healthier plants. So the other thing is with seed collection, it usually is from mid-June to late October. And usually you can get out there pretty much 3 or 4 days a week for like half a day. So it can get quite fun doing that. Most everybody, that's the job that they want when they come to work with me. So the timing of collection is pretty important. You also not just because of weather variation, everything, you can't just say, okay, last year it was June 22nd when I collected cut leaf daisy. It could be different because of weather conditions. So let's see. The other thing I was going to talk about was you also have to factor in there's early, mid, and late flowering, which corresponds to early, mid, and late ripening. And you're going to get the most seed mid, but if you miss it for some reason, you still have a chance to get early and late. And My preference is to try to get a little bit early, mostly mid and some late, because once again, genetics. You don't know what's going to have the best genetics and survivability. So one thing I always think about is increasing the genetic diversity. And usually when seed ripens, it's about 4 to 6 weeks after it flowers. Also, once again, temperature dependent. It can be a little bit dependent on genetics, because that's how you get your early, mid, and late flowering or ripening. But it also can be dependent on wind, cold, rain. All of these factors can have an effect on when it's going to ripen. Okay, so my collection strategies for Erigeron compositus, it's a very early one. It's one of the earliest ones. It's actually pretty easy. You just pull the little head off when the seeds are ripe. That's all you really have to do. It's quite easy. Now, the puccoon or the Lithospermum ruderale is a lot more complex. What you do is you can, there's a lot of different ways you can do it, but the easiest way is to put a tarp around it and then beat it, and the seeds fall off. And because it has very prickly hairs on its leaves, that's the way you really want to do it, because otherwise your hands get really itchy. Okay, now we're on to seed cleaning. And this usually happens late fall. As you saw before, as I told you before, seed collection runs till mid-October, and then after that, except for the berries, which get done almost immediately after they're collected, seed cleaning happens later in the year. There's our stuffed refrigerator, and there's some of the seeds that we have bagged. So this is some of the equipment for seed cleaning. We have a tabletop clipper, we have a blender, we have a scale, and we have some sieves or screens. And the clipper, so all of these, the blender actually we use quite a lot. It's not just for berries. For smaller, very tiny seeds, it works really well to beat them out of the seed, the pods. So there we go. So there's some more equipment. We're a small scale operation, so we have the small tabletop models and everything. But if you were a bigger operation, you would have a big air separator, a dib vig, which is how you would clean large amounts of berries, a debearder, which works really well for beating the pappus off of seeds, or and then a fan mill. And you can see how large those guys are. That's a commercial operation producing a lot of seed. So when we clean seed, it's a multi-step process. And the process is different for each species. So one of the first things we do is, if there are any large stems, we get rid of them, because if you chop up the stems or somehow the stems get smaller, they actually weigh a lot like the seeds, and then it becomes really hard to separate them. So the first thing we do is make sure there are no stems in the seed cleaning process. And then from there, you can use a blender, or you can see Mark Mariano, he is rubbing to get the pappus off the seed. We don't have a debearder, so we use our hands. Once again, for seed cleaning, hands are pretty important, since we don't have all the variety of equipment that a commercial operation would have. Okay, seed sizes and weights. By the way, this is pretty much how clean we want to get it. The more clean you get it, the less chaff you have, the less disease that you're going to bring in if you have to treat the seed, or even if you just sow the seed dry. And as you can see, people, I put this in just because people always they don't estimate the amount of small seeds that you're going to need when you're either sowing seeds out directly onto the ground, or even if you're putting them into a tube to get them to grow. Let's see.
What seeds are these? Can you tell?
Yeah. The top one is Doug fir, and the bottom one is common evening primrose. And when we do the estimate, my example is Artemisia tridentata. It's extremely small, and if you were just wanting to do a test in a meter square plot, 100 seeds doesn't sound like a lot, but you have to think about when the plants grow up, and usually you get really good germination with them. So you wouldn't want 100 seeds in there. And if you overestimate what you think 100 seeds is, which is just a speck, it's not even a pinch, it's less than that, you can see how easy it is to overestimate. I do it, everybody does it. I also wanted to say, not all seed is good seed. You can have a lot of things go wrong with the seed. If due to environmental factors, they can produce a seed without an embryo, and that's an empty seed, and that could be due to not getting pollinated. That could be due to any number of environmental factors. I know that this past year we didn't have such great seed for the early stuff because it got really cold, no pollinators were coming out. If it's wind-borne, if it rains, the pollen is not going to travel as far. And so there's a lot of ways that you can get not good seed. And some seed, I know back in the bitterroot, antelope bitterbrush or Persia tridentata actually has multitude of diseases within the seed, within the embryo and the endosperm itself. And when I was working back there, we would try everything to get it to grow because it's a really nice plant. And finally we gave up with collecting in the bitterroot, and we outsourced it to somewhere else where it had a cleaner seed, because even when we treated it with fungicides and stuff, it still died. Okay, so we do seed testing. We do germination tests, which is where you take 100 non-dormant seeds, meaning that they will germinate if you seed them, if you just put them in a petri dish with some water. And then you just count how many of those 100 seeds germinate. And what that test helps with is knowing how many seeds you need to sow in a tube, because seeds do cost money, and you don't want to oversow. You also don't want to have to thin those tubes out, because once again it takes people time, and that's money. We do cut tests on everything that it has a dormancy in it, and that just tells us if they're empty or full, which can be helpful. The one test we don't do is a TZ test or Tetrazolium test. The reason we don't do it is because Tetrazolium is carcinogenic, mutagenic, teratogenic, and it's not something you want to mess with. You have to have a nice, well-equipped laboratory with fume hoods and stuff like that to actually work with it. But that is actually also the best test to tell you how viable your seed is, because even though a seed has dried down, it still is a living thing, and it does slowly respire. And what a TZ test does is, if the tissue does respire, is living, it turns it red. Anything that stays white is dead tissue, and that can tell you really well what kind of germination you're going to be able to get. Okay, so cleaning strategies. We have on this side allium and that side Artemisia tridentata. And you can see the difference in sizes. The nodding onion is pretty easy to clean. Once again, you just take the stems off and you can just rub in between your hands to get, they have a papery enclosing the seed. The seed head has a papery, I'm forgetting the name now. But anyway, so you just have to rub it, the seeds come out, and then you can run it through the clipper, which uses air to separate the seed from the chaff. Artemisia tridentata is a little bit different. What you first have to do is you cut it off with clippers, so you have large stems in it, so you have to pull it off the stems. Then what you do is you use a blender, you blend the seeds out and then once you have blended it, you take everything and then you use a clipper to separate it using air. And you can get it, these are really pretty clean. We always try to get at least 90% clean without too much dead plant material in there. Okay. Now the next step in everything is seed treatment. And this is, we're doing imbibition, which means we're getting the seeds to imbibe water. And if you have a lot of seeds and you have a larger container, one of the things that you can do is if you have a lot of water, you can drown the seeds, which is why you use a bubbler or an aerator to make sure there's enough oxygen in there, because besides needing water, the seeds also need oxygen to survive. And so usually what we do if we don't have a lot of seeds and we don't do large amounts of seeds, we usually just use a petri dish which has a large surface area and a small amount of water, and that allows for gas exchange quite readily. And so we don't always need a bubbler for everything. So here's some of our equipment. Beakers, graduated cylinders, pipettes, strainers, bubblers, and then so with seeds what you want to do is you need to clean the outer surface of the seed, and so we use bleach and hydrogen peroxide. Some seeds there are different types of dormancies, which I won't get into here because you get more complex, but one of the things that if an embryo is small, you can use gibberellic acid to help it grow. And then, of course, petri dishes. More equipment. You can use, there is one technique, scarification, and that's where you would use sulfuric or phosphoric acid to scarify seeds. Rock tumbler is a new technique that they're starting to investigate and use with seeds that need to be scarified. The reason that they're using the rock tumbler method is because, once again, sulfuric acid, phosphoric acid is very detrimental, and it can't, when I used to do sulfuric acid treatments, I would wear a rubber suit, green, you'd have your hands covered up to here, you'd have the big coat on, you'd have the pants with suspenders, you'd wear rubber boots, the coat would go up over your head, you'd have a respirator mask on, and then you go out and treat seed. And, so the rock tumbler is a lot less caustic, a lot less toxic. Yeah?
What molarity was the sulfuric acid?
H2SO4. Or molarity. Okay, that varies. One of the things that using sulfuric acid, you have to be really careful because, and maybe I don't remember, but when we were doing it, back then, a lot of it's temperature dependent because the reaction is so much slower with cold weather, and it's so much faster with hot. Does that kind of answer your question? I mean, I'm sorry, I don't remember.
Is it concentrated or dilute sulfuric acid?
We had a big barrel of concentrated, but we would dilute it, and the dilution would be dependent on the seed.
About 1 to 10, did you dilute it 1 to 2? Did you?
Well, I mean, if I had to guess, which I am kind of, probably 1 to 10. It wasn't.
That's what I remember too.
It wasn't, but it did kind of vary with what seeds you were treating. So there's a lot of variation in that.
Thank you.
Sorry, I don't know better. And then, of course, one of the newer things that I've been looking at, and I just tried last year, was using liquid smoke. And the reason people are now talking about liquid smoke is they used to do smokers to get some seed, especially in California, where there are a lot of wildfires. And so the smoke actually does have an effect on germination. A lot of seeds that are fire dependent they're finding are actually smoke dependent too. So, okay. So techniques. One of the techniques that we were just talking about was scarification. The tumbler, I have done a little bit of scarification, and not recently though, just because I don't have a lab with a fume hood and all the protective equipment. We have tried using sandpaper. It does help. It is not the best. Some seed coats are really brittle and crack easily, and so sandpaper doesn't always work. Then we do a surface sterilization, and the reason we do what I call surface sterilization, and that's where we use either bleach or hydrogen peroxide diluted. In both cases is because when you take seeds from the wild, you've got bacteria and fungi coating the seed coat. And what you have to do, because if you're treating them in some way, and then you put them into coconut fiber and put them in the refrigerator to do stratification, it's kind of a really good way to promote growth of bacteria and fungi, which can kill young seedlings. So even though there are sites out there that tell you how to germinate native seeds, they almost never say surface sterilize. And one of the things that I always add in is surface sterilization, just to make sure that I give the seeds the best chance they have to germinate and survive. And then we imbibe them with water. And that is a lot of techniques don't tell you to imbibe. They just tell you to put them in the refrigerator in moist cocoa fiber or peat. But I've found that if you imbibe them for anywhere from 2 hours to 72 hours, depending on the species, it helps jumpstart, and you actually can cut down the time that they're in the refrigeration in stratification by quite a bit. And then stratification. So stratification usually means cold, and basically what you're trying to do is mimic winter conditions to get the seed to germinate. And so we stick them in. So once again, we stick them in plastic bags, but the plastic bags have small holes, and we keep the top open. The peat or coconut fiber stays moist. I find coconut fiber is better just because I find it doesn't allow for as much fungal growth or bacterial growth. And so you keep that moist, you keep them in there, and then it actually cuts down the time rather than just sticking them outside in tubes. It can cut down your time. You also have a lot more control over environmental factors if you do the refrigerator method. Okay. So more techniques here. We do cuttings. We usually try not to do too many cuttings. Once again, it's labor intensive, which means it's way more expensive than growing stuff from seed. But there are species that are recalcitrant, meaning that nobody has figured out a good way to get their seeds to germinate. And one of those species is juniper. The newest technique I saw for getting juniper seeds to germinate takes 2 years. And nobody really has that time. So what we do is we do clones, cuttings, which are clones of one plant. So once again, thinking about the genetics and genetic diversity, you try to get from as many different plants as possible. With junipers, you do stem cuttings. They have to be a certain size and a certain length. Usually it's you don't want big, tough, woody stems. You want smaller, not quite at the tip, but they're about pencil-sized, usually is the diameter that they're thinking. And they're only about 3-4 inches, 3 to 6 inches in length. You don't want them longer than that. What we do is we then use IBA, which is a hormone that makes the cuttings want to root, and then we stick them in sand perlite mixture, and we stick them under a tent where they can get as much moisture as possible. We keep them really moist. One of the things that we're finding out is you can't take the cuttings at any time of the year, and so it's very seasonal. Okay, we're getting close, everyone. So seed treatments, they're very different. The lupin just needs a lot of water. It's a very big seed. So what I do is I give them as much water as they can possibly handle, because they're going to take so much water. I get a lot of questions about why aren't my lupin seeds germinating? It's because if you just throw them on the ground, they need a really, really wet spring for them to germinate. So I don't just throw them into a tube and hope they germinate. They have to have, we put them on tissue or paper towels, and we soak them. And that technique seems to really work. Cercocarpus needs a stratification. So I'm not going to talk about that too much, since Robert told me I'm talking too much. Seed sowing. So the equipment: trays, tubes, spoons, chopsticks, yes, tweezers. Chopsticks actually are really good for making holes, or if you're sowing dry seeds, you can easily flick them off with chopsticks. So I'm always on the lookout for chopsticks. Filling trays. It can get very complex, especially if you're using mycorrhizae, which is what we are doing. So I have a little sign that all my students look at before they start filling trays. And there's Joao doing just that. More techniques: how you sow dry seed, how you sow stratified seed are very different. And then we also do some transplanting. Once again, these are all. They have similarities, but they're definitely different, and each has their own technique. And then sowing strategies. Little flower penstemon, sorry, that's not the best picture. I had to blow it up because otherwise it just looks like dust. And then Gaillardia. What we do is, that's another one that likes to be soaked in a petri dish on paper towels. These guys go outside, penstemons. That's because they have an even more complex stratification than just sticking them in a refrigerator. They probably need light or variable temperatures to get them to germinate. Seedling growth. Equipment: tables, grow light, hoses, sprinkler system. There's one of our grow lights. Watering. Okay, watering can be very complex in the greenhouse, in our staging area, which is where we keep our plants over the summer on site. And then there are seasonal factors. So the one I want to talk about the most is seasonal factors. The big thing is to remember that in the fall, getting towards fall, you want to start cutting down on your watering. You have to help make the plants go dormant, because if you have a really sharp cold spell, it can kill the plants. Okay, mycorrhizae is a fungus. The two that we're mostly concerned with are ectomycorrhizae, which are for the conifers, and endomycorrhizae, which is for most of the other plants. It is mutualistic for the most part, and it's found in 95% of the plant families. It colonizes plant roots. Let's see. So I will, if people can ask me questions about it later if you have more questions. Fertilizing, organic, synthetic, that doesn't bother me as much as, because we're putting mycorrhizae into the soil, the big thing is how much phosphorus is in our fertilizer. When you have mycorrhizae, to get them to colonize a plant root, you need to have a low phosphorus. 1, 2, or 3 is what you want to have for phosphorus, percent phosphorus. Because what you want to do is you want to force the plant, because plants can't use inorganic phosphorus, you want them to be colonized by the mycorrhizae, which can actually utilize inorganic phosphorus. So that 3% is for organic phosphorus. Hardening off, you do that in the spring when you take them out. It can get complicated because, of course, here it loves to snow in June, and that can be pretty hard on the plants. So it's always a nail-biting time when we move plants out of the greenhouse because they're still very tender. In autumn, we winterize, get everything ready for winter. And another really scary time is overwintering, because it's so easy to lose plants over the winter. Because you have to be careful about how much you water, when you water, what the temperature is when you water, and you have to keep them dark enough, and then you have to hope that you don't get a winter with fluctuating temperatures, because what kills most plants is not the cold, it is actually fluctuating temperatures, because once they get to a certain point with the cold, if it warms up after they've had the amount of cold they need, they will start to grow again. And you don't want that. And that is actually what can kill a lot of plants. I'm just going to skip over these. This is a forb, and that's a grass. They have a little bit different growth strategies. Planting on site. Here's a picture of one of the places we planted. Equipment. Oops. Hand trowels, power drills. Power drills are really great. I don't know who thought of that, Chris. You maybe could tell, but they work really well for doing planting, where we're doing planting. Shovels, rakes, and gloves, also good. Techniques. One of the important things is getting the correct depth of hole for the plant. You don't want the roots to turn. You want to keep them going straight down. Rocks are also good. New technique that we're using is rough and loose, where the planting area is not flat level. You have some dips to help catch the rainwater and give the roots a little bit of shade. So more techniques. This just shows you that you can have a really steep slope, but you can have some plants growing on there, and you can see that they've spaced themselves out to where they need to be. So, but anyway, the correct species at the correct site, slope, aspect, and elevation are all critical. Okay, and more techniques. This is if you want to know more about GIS modeling, you need to talk to Mark Mariano. But I will try. What he's doing is, I hope I get this right, is you take factors like aspect, elevation, slope, and you put these layers over a map of, say, butte, and where they meet for a certain species, and that species needs like northwest slope, steep, and at 54 elevation. When you layer those over, his GIS modeling will tell you exactly where you need to plant that species. Okay. Then planting strategies, you can see we do rocks, we do water, we do everything in between. Thank you to Robert Powell. Most of those pictures are his. Thank you to Mark for his picture. Rachel, who's one of our students too, besides Mark, took some pictures. I want to mention Matt Ogden because he is my native plant growing guru. He taught me everything I know, so if I get anything wrong, it's not him, it's me. Then the BNRC and the NRD for funding us. And any questions? Yeah.
Okay, so I have 2 questions.
Okay.
With respect to so there is all these nice native areas, okay?
Yeah.
And when you're doing the germination,
Yeah.
and presumably these plants are evolved so they can germinate and propagate out there in the
Yes.
world without humans intervening.
Right.
Okay? When you talk about using things like bleach and sulfuric and phosphoric acid, I'm wondering what it is about where they're supposed to be living and growing that this rather abusive sounding treatment is emulating or helping them do it in captivity, what they would have otherwise done
Okay.
in nature.
Well, one of the things, so when we do outdoor stratification, we just put dry seed in and leave them. One of the things that outdoor seed has that if you're in the refrigerator doesn't have is UV light.
What?
UV light. UV light is a great sterilizer. It will sterilize those seeds and keep a lot of the fungus or the bacteria down to where the plant can grow. And the other one you were talking about was...
I said bleach, sulfuric, and phosphoric acid. I was wondering.
Okay,
Those
so...
seem pretty nasty to me.
So actually, a lot of the seed gets eaten, and it passes, or the berries or the fruit gets eaten, passes through the digestive system, and we've got a very caustic stomach, which actually people have given me seed that has gone through birds and bear poop, and it germinates beautifully, absolutely beautifully. Don't have to do anything to it. So I think that is where that's coming from, and so we have to mimic it some way.
Okay. And then the other question is, in these different types of species, what sort of percent germination do you get on the seeds? Is it a very high percentage or a very low percentage?
You know, usually if we do cleaning right and we have a good year, we get pretty high percentage, but it is variable dependent on a lot of environmental factors from when the seed was growing or being fertilized, and then how well we clean it also has a factor, because there are a lot of empty seeds that if you look in the aster family, they're notorious for producing a lot of empty seeds, and so it's also how well we clean it. So it is variable, but usually with the techniques and the stuff that we're doing now, it's usually pretty high. Anyone else have a question?
Is that a bear that's trying to eat the seeds?
That's some of the stuff that we have to deal with when we're out there.
West of that, actually.
Yeah.
The area is still to be known.
Yeah.
So you mentioned issues with genetic diversity.
Yeah.
Have you tried doing cross-fertilization or doing the fertilization, moving pollen around?
No. one of the things I maybe didn't emphasize enough is, you know, that would cost money because it'd be hand labor, and we don't have the manpower.
Population.
Yeah.
Good genetic diversity. It might
Yeah.
be worth it.
Yeah. Yeah, I mean, there's a trade-off there. It is something you have to consider.
Yeah.
And especially here where you do, for many species, have lower numbers than you normally would have. Any other questions?
Thank you, Krystal.
Okay.